So today we're going to cover DNA replication by Tamar and Gigi. First, we're going to start with the chemical structure of deoxyribonucleic acid, also known as DNA. There are 4 bases total. Here we can see the structure of adenine and where it is located on the double helix. Next, we have thymine, which is complementary to adenine. They will always complement one another. The other complementary pair is cytosine and guanine, which we will see the structures and positions of shortly. As you can see, the structure of thymine resembles cytosine, and now you can see guanine, and you can see that adenine structure resembles guanine. We will talk more about why that is later on in the video. But yeah, they always complement each other, A and T, C and G. To continue, DNA is a polymer made up of units called nucleotides. Nucleotides are made of 3 groups: deoxyribose, which is a sugar group, a phosphate group, and a base. Here you can see the sugar-phosphate backbone with all the parts, all the groups. These strands, backbones, are held together by hydrogen bonds. These bonds are located between adjacent strands. Again, A always with T, C always with G. So here we have a phosphate group connected to a deoxyribose group connected to a base. And A is bonded to T with 2 hydrogen bonds, while C is bonded to G with 3. So here's some basics. And DNA uses anti-parallel strands. 5 end of one strand is always parallel to the 3 end. This is because if you use anti-parallel strands, you can have complementary bases. Again, A always with T, C always with G. So back to the bases. The reason A and G are similar is because they are both purines, meaning double ring structures. And T and C are both pyrimidines, meaning single ring structures. Purines always bond with pyrimidines. Now we're going to cover some basic enzymes that play a role in division and replication. Here is primase. It's an enzyme that synthesizes short RNA sequences called primers, which tell the DNA polymerase where they have to start building. Next, we have a single strand binding proteins, or SSP for short. After the enzyme helicase separates double helix, those strands want to reform that helix, but SSP prevents them from doing so. It keeps the strands open so replication can occur. Another helper enzyme is topoisomerase. I have no idea how to pronounce it, but it unwinds during replication. The winding problem of DNA arises due to its structure. Okay, so step 1, helicases unwind the double helix. It basically just unzips it like a zipper. Yeah. Step number 2 includes the SSBP proteins we talked about. So these stabilize the unwound DNA from reforming a double helix, which is the strand's natural thing that they want to do. They want to reform the helix. Step number 3, 2 leading strands, one for each strand of DNA, are synthesized by the enzyme DNA polymerase in the 5-3 direction. Now 4, short DNA primer synthesizes the lagging strand in the same direction. The main difference between leading and lagging strands is the continuation of synthesis. Leading continuously synthesized, while lagging discontinuous synthesized, meaning they have short sections, which are called Okazaki fragments. The next step, and the final-ish step, is once the RNA primer is removed, because it's no longer needed, DNA ligase joins the Okazaki fragment to the growing strand. So it basically bonds it back together. There are leading and lagging sections of replicated DNA on both strands, but we only show one because there's not enough room to draw them all. So here's the overview of all the steps, and you can basically see how they play, but they do not all act at once. Clearly, they go in the order of the steps. So while you're watching this, a quick DNA polymerase definition said there are enzymes that synthesize DNA from deoxyribonucleotides, which are the building blocks of DNA. DNA ligase joins Okazaki fragments by closing nicks in the linkage between 3-carbon atom of one sugar molecule and the 5-carbon atom of another. That is basically all you need to know, and I hope you learned a lot from this video.